Hi everybody, it's your AP Biology teacher, Mr. Poser. Today we're getting into topic 6.2, and that is on replication. So this unit is on gene expression, and I talked about DNA and RNA structure in the last topic. And I talked about how important DNA and RNA structure were going to become for our understanding of how gene expression works, right? And gene expression is the process by which cells make their proteins using DNA as a code, right? But we're not getting to there yet. We're getting into replication, which is if you take apart that word replica, a replica is like a copy, right? So you're copying DNA. Hence what this image is about down here. So we are copying DNA today. We're not really doing anything with RNA or with proteins just yet today, or newly synthesized proteins today. We're going to get into how DNA is copied. So in our 2 units ago, when we talked about the cell cycle, all right, we talked about how a cell through mitosis is able to, copy all of its DNA and split into 2 genetically identical cells. Well, that's not possible without DNA replication, and this is a super important concept, and it needs to be talked about in a biology class because why? Well, it ensures the continuity of life on Earth, right? So it ensures that, not everybody can live forever, right? So you got to pass down your genes, right? If to permeate your species, right? In order to pass down your genes, you have to copy your DNA. Hence, DNA replication, right? So as I put up here, for DNA to be passed down to offspring, it must be copied. Replication makes sure offspring have genetic information. Here's a little schematic of mitosis once again, just as a reminder, right? You're making 2 identical cells, and how do you make identical cells? Well, they got to have identical DNA as well, because that's the code for making proteins, right? We're going to talk about. So replication is going to become extra important here. It just is extra important because it ensures that life on Earth continues, right? So here's the deal. I'm going to animate kind of what DNA replication looks like. I'm not going to put a whole lot of text on the screen, but I am going to kind of explain this as it goes, and then all the text that's necessary I put in the recap at the end of the video. So if you're taking notes, right, you're wondering, oh, what do I write down? It's at the end, all right? I just wanted to keep this animation here because it's easier to visualize, I think. But here's a good DNA molecule. Well, it's 2 lines, but it's a DNA molecule. It's representing a DNA molecule, all right? And you can see, once again, we have a 3' to 5' strand, and we have a 5' to 3' strand, all right? And if you remember what those mean, this is anti-parallel. These 2 DNA strands are facing opposite directions. They're kind of upside down to each other, and that's going to become pretty important here in just a second here. Why that matters. So check it out. In order for DNA to be copied, the first thing that has to happen is that it needs to be split apart. All right, those base pairs, the A's and T's, apple goes in the tree, car goes in the garage, right? Those hydrogen bonds holding those base pairs together need to be separated out. All right, so those nucleotides can be kind of accessed here. Okay, so we're not going to make a new DNA molecule just by taking an entire DNA molecule and making a copy of it. Well, we're going to make a copy of half of it, 2 copies of half of it, if that makes sense. And you'll see what I mean here in just a second, all right? But they're going to be split apart first, all right? And then I don't have it labeled right now, but we have some special enzymes that are called DNA polymerase, all right? And DNA polymerase is going to slide along what are called these template strands, these old strands of DNA, and it's going to make new strands based off of the base pairing rules, all right? So here's DNA polymerase. There it goes. They go in opposite directions because it only works in what's called the 5' to 3' direction, all right? So that means here's the 5' end on the new strand, the 3' end on the other. It's going to move this way on this strand and move the opposite way on the other strand. Okay, and it's going to make new DNA strands based off of the template strands, right? So let's just pretend, all right, let me see where my cursor is. There's an A right here. That means that there's going to be a new T nucleotide that's across from it, or a thymine, right? Here's a cytosine, or a C. There's going to be a G across from it. Here's a T. There's going to be an A across from it, so on and so forth, right? And if you do it correctly, that means that this newly synthesized strand, all right, since the base pairs always match up and it's always going to be, the same pattern, this new strand is going to be exactly the same as this old strand over here, all right? It doesn't matter, right, whether They're split apart, and they're going to be the same either way because of the base pairing rules, all right? So here's my 2 new strands of DNA. Now, how this process is described, as I said before, it's in the 5' to 3' direction, right? So they're going whoop, opposite ways. DNA replication is semi-conservative, and that is not a political stance, right? So semi means half, conservative means conserved or saved, right? In my 2 DNA molecules that I now have, they're made of 1 strand that is conserved, that's left over, or the old strand, and 1 newly synthesized strand, okay? And this is going to happen every time DNA gets copied. This happens again and again. 1 strand is saved, 1 strand is new. 1 strand is saved, 1 strand is new, or old and new, in other words, okay? And that's what semi-conservative means, all right? And it took a whole bunch of amazing science experiments for us to figure out, oh, DNA's replication is semi-conservative, because scientists weren't sure for quite a while how DNA gets copied, all right? So there it is. It is semi-conservative. Now, we're going to get into a few more details about how this works, particularly talk about some of the functions of the enzymes that are responsible for DNA replication. And there are four, I believe you have to know, for the AP Biology curriculum. And the first of which is helicase, right? So you'll see, as opposed to the model that I just had on the previous page, there's kind of like this, angle to it. All right, this is what's called the replication fork over here, and it's not like a fork like you eat spaghetti with. All right, it's like a fork in the road, right? It's where these 2 template strands are split from one another, and the enzyme that's going to be doing the splitting, and I like to use a triangle to represent it because it kind of like wedges between the 2. It's called helicase, right? DNA is a double helix. Helicase separates the helix, right? So that these two sets of nucleotides are exposed, right? They're separated from their partners. Oh, they're so sad. All right, but they're gonna, well, actually, yeah, they're never gonna come back to each other because they're gonna get new partners. But anyway, helicase is splitting apart the DNA strands from each other. All right, and when you split apart these DNA strands, remember, DNA is a twisted ladder, right? It's all twisty shape. What's going to happen when you split these apart from each other? It's kind of like pulling apart a piece of yarn, right? The other end, basically upstream of where you're pulling apart those 2 strands, it gets extra twisty. All right, and the word I just used, upstream, just means basically like beyond the where it's being split, beyond the replication fork, right? But it's going to get extra twisty up here. Whoops. Sorry. It's going to get extra twisty upstream of where it's getting split, unless there's with this enzyme called topoisomerase, which prevents the formal definition is that it prevents supercoiling, right? Because that can cause problems if it's too wound up, right? And it's not going to be replicated properly. So topoisomerase kind of smooths it out a little bit. All right, and so once these are separated, all right, what's going to come next is that, all right, we got to synthesize the new strands based off of the template strands, right, based off the old ones. But there's one other thing that needs to happen before that, right, and that needs to be the placement of what are called primers. And primers are little tiny sections of RNA, actually, that serve as a starting point for DNA polymerase, all right, and we're going to show you where DNA polymerase comes in just a second, right, but basically these enzymes called primase place a little bit of RNA, and that provides a starting point for DNA polymerase to bind to. All right, DNA polymerase is an enzyme, which means it has a particular shape that allows it to do a very, very specific job, right? And DNA polymerase is so shaped that it can't bind to the DNA template strand without a primer there for it to bind to and start to work off of, right? So prime means first, right? Primers have to be placed first, and then DNA polymerase binds to the template strand and then does its thing, right? So this DNA polymerase is going to move in this direction, right? Here's the 3' end of the template strand. That means this is the 5' end, and this is going to be the 3' end over here because it's anti-parallel once again. So DNA polymerase is going to work this way on this side, but it's going to work this way, the other way, on the top strand over here, all right? Because look, here's the 3' end of the template strand, here's the 5' end of the template strand, which means this is the new 5' end, and this is the new 3' end, all right? So this one's going to move this way, and this one's going to move this way. All right, that's great. Awesome. All right, so let's see that happen. I know my animation is not great, but, it gets the job done, I hope. All right, but DNA polymerase slides along those template strands, and it adds new DNA nucleotides based on the base pairing rules in order to make new DNA molecules. All right, great, cool. All right, but it gets a little complicated. Now, And that has to do with DNA being anti-parallel and DNA polymerase only working in the 5' to 3' direction. All right, there's a little bit of an issue there. It's not a huge issue, okay, because it's easy to solve here, but it complicates things a little bit. All right, now helicase is moving that way, right? It's moving upstream. All right, and this DNA polymerase down here is moving in the opposite direction as the helicase, and this one is moving in the same direction as helicase. All right, so I'm going to try and visualize this here for you. Okay, if I'm getting rid of the other stuff and I'm trying to make it clear here, look, the helicase moves that way, and this DNA polymerase on the top strand is able to just kind of follow along with it, but the other DNA polymerase has to go backwards because DNA polymerase only works 5' to 3'. Okay, so you kind of have a disconnect here on this bottom strand. All right, as helicase moves in the opposite direction, while DNA polymerase is going this way. All right, but what about all this DNA that needs to be copied over here? Well, you got a big gap. Well, what do you need to do? You need to build this DNA strand a piece at a time. So you need to place another primer, another DNA polymerase, and have it work backwards again to make up the gap there. Okay, so this bottom strand has to be put together piece by piece by piece by piece, but this top strand, all right, DNA polymerase can just slide along through and make DNA in one continuous strand. Okay, but this bottom one that has to be built a piece at a time is what's called the lagging strand, all right, because it has to work backwards and it's going away from the replication fork. While this top one is called the leading strand and it can just go towards the replication fork. The lagging strand is built piece by piece by piece, and leading strand is just all one fell swoop because it's working in the same direction as helicase is. All right. So that's the difference between lagging and leading. All right, and last thing here, a last enzyme that I got to talk about is this thing called ligase. You might have seen that on your screen just now. And ligase is going to replace the RNA primers with DNA, and it's going to piece together these segments here. All right, this especially on the lagging strand. Each segment has got to be sealed together to form one continuous strand. So that's what ligase does. It flies through here. Oh, it doesn't fly. All right, but it slides along those template strands and pieces those fragments together. Okay, so here's the recap. Here's all the text that I was talking about. I just kind of left the animations on the screen. So if you're writing notes, this is what you're writing here. Ready? Replication is the copying of DNA that allows organisms to pass down genetic information, and that ensures the continuity of life, right? It makes sure that cells have, their offspring have genetic material to work with. And it should be the same genetic material if we're talking about mitosis, right? Replication is semi-conservative, not a political stance again. It's the fact that a new DNA strand is built off of a template strand, and each new DNA molecule contains one old strand, one that's conserved, basically the template strand, and one newly synthesized strand that was built based off of the old one, based on the base pairing rules. Apple goes in the tree, car goes in the garage, right? DNA is replicated or copied in the 5' to 3' direction. DNA polymerase can only work in the 5' to 3' direction, and that complicates things, as I'll talk about again in a second. But let's talk about these enzymes. What do they do? Helicase is the enzyme that's going to separate out the template strands, split up that double helix. Topoisomerase is the enzyme that's going to be preventing what's called the supercoiling, all right, making sure it doesn't get too twisty upstream of where the replication fork is, basically where those two strands are being separated. Primers, before DNA polymerase does its job and makes a new DNA strand, primers need to be placed by an enzyme called primase as a starting point for DNA polymerase. It can't bind to the template strand without a primer. Okay, and then DNA polymerase synthesizes new nucleotides. It adds an A for every T, it adds a G for every C, so on and so forth. Synthesizes new nucleotides that match with those on the template strand based on the base pairing rules. It goes continuously on the leading strand because it goes towards the replication fork and is moving in the same direction as the strands are being separated. And in segments on the lagging strand, it has to go piece by piece. Go back, start over, go back, start over, go back, start over on what's called the lagging strand, all right? And so it's built in segments. All right, and ligase, which is the last enzyme here, its job is to seal those segments together on the lagging strand and the leading strand. All right, it replaces the primers to form two continuous strands of DNA. All right, so that you have a new molecule that's ready to be, used. All right, that is it for this video. We're going to get into some gene expression in the next videos. So stay tuned. Let me know if you have any questions. We'll see you next time.